So our next speaker is Dr. Ashok Chanda. Ashok's spoken here many times, so most of you know him. He's going to talk about some of the disease research that he's been working on.
Thank you, Todd. Good morning, everybody. Just trying to see my here. This, oh, perfect. Thank you. Well, I was talking to somebody this morning. This is, to me, is like a season kickoff, right? So I think it's great to see everybody just try to learn from each other. And I think the goal for me for today is just to talk about, spend some time about rhizoctonia management. It looks like it's day in and day out we are dealing with rhizoctonia. And then talk about some of the uncommon diseases that we're seeing in few fields, but they're not major problems, but especially keeping our industry folks in mind and some of the crop advisors and crop consultants. It's nice to get some of these things under your radar. You never know what you'll find in your field, right? That's how your day goes sometimes. So again, why do we need to know what we are dealing with in a particular field, right? So for this particular example, this is back in 2015. We thought we were just dealing with rhizoctonia in this particular field, but look at the date there, right? By July, most of the stands were gone. It's not just rhizoc, it's a combination of Aphanomyces and rhizoctonia. That can be very severe, right? So I think by the time a couple of weeks later, there's nothing left in this particular field. So that's very critical for this. So looking at the samples, I'm only showing the data for the disease samples that we got in 2023. As we can clearly see, rhizoctonia is again clear winner this year, and then followed by some fusarium. Most of the samples came from up in the valley. There may be a couple of samples from here. Again, fusarium is something we have to be very careful when we have in the field. So thinking about rhizoctonia, I showed this slide before, right? So it can actually kill the seedlings before or after emergence, right? Pre or post emergence damping off. And also some of the younger plants are more susceptible to rhizoctonia compared to the older ones. So we have to be really careful for this one. But as you go into later into the season, sorry, next one. Can I actually control this here or here? Okay. I think I'll take care of this here. Thank you. Oh, it just keeps going. So how do we go to the next one here? Okay, now I see this. Sorry. So as we go later into the season, you can actually see that some of the chlorosis there. You lose a lot of plants there, like David showed. As you go later part of the season too. But by the end of the season, all you see is spiders, right? So most of the roots are dead, and these petioles are just thin and wiry. So that's all you see. And typically, by the time we go to harvest, now this is what we are seeing, right? So the typical rhizoctonia, we see ladder-like pattern, reddish-brown lesions, but then this is also not uncommon for us, right? The top of the root looks clean and the really bottom portion, but you can have rhizoctonia here. Again, if you're not doing any cultivation, you're not throwing any soil, so most of the rhizoctonia is down here, right? When these roots die, this is what it makes. This is how rhizoctonia looks on the plates. But exactly, these look like just like soil particles. There's no way for us to know it's soil or rhizoctonia. So it can survive in the soil for 2 to 3 years. And good examples is some of the samples that are received from your fields here, right? So this is about from 4.5 weeks to 7 weeks to 9 weeks, right? These are looking very similar to what I showed from our inoculated trials, right? Again, rhizoctonia comes a little bit later in the season. I think in the past we talked about, maybe having that N4O fungicide at the time of planting followed by post-emergence, but I'm going to show some data in the next few slides here, right? When we think about rhizoctonia, like predominantly for the anastomosis groups 2-2, 3B, and 4, right? These are the major ones that we are dealing with in sugar beets, but for you guys, it's 2-2, 3B because it likes to grow at a little bit warmer temperatures, compared to us up in Crookston, it's 2-2, 4. And then, like I said, the sclerotia, the dormant structures can survive in the soil for 2 to 3 years, right? So if you grow sugar beets and come back, in the year 4, you can still expect some rhizoctonia pressure. But the real problem, if you have soybeans, corn, or dry beans in the rotation, these are also good hosts for rhizoctonia. So you're not really getting rid of rhizoctonia in any given year. So that's why it's always, it has to be on your mind just how do we manage rhizoctonia, and take notes every year to do this. And then the big difference between rhizoctonia and Aphanomyces, it tends to be very patchy. You can hit a patch of really bad rhizoctonia in a field, and right next to it, it looks really fine, because that's how rhizoctonia grows and multiplies in a field. I think, it just, once it reaches a critical mass, it starts killing these plants. Whereas Aphanomyces, it's present more or less uniform in a whole field. And then depth, right? When we talk about, where do we see rhizoc? It's mostly in the top 4 inches. And in some fields where we are seeing severe root rot, by the end of the season, we are seeing all the way up to 6 inches. But most of the rhizoc inoculum is in the 4 inches, top 4 inches. It's a good example that soybeans, the same thing happens. Before they emerge out of the ground, they die, the seedlings, you will not see anything there. And then typically you see reddish-brown lesions, on these dying seedlings there for soybeans. And navy beans, again, these are inoculated with the same rhizoctonia that we use for the sugar beet trials. You see a row of plants dying. Again, the younger plants that are emerging a little bit later, these are more susceptible, compared to the other ones which are real strong there. And what else will impact rhizoctonia, right? So the other trial that we looked at at Crookston was actually the drain tiles were installed back in 2001. We have it spacing from 15, 25, 40, and 60 feet. The one that's 0 says it's no drain tile for this one, right? So we're looking at a vibrance seed treatment, the same seed treatment that's available for sugar beets, and we applied Inforo Quadris, and also this is my check, non-treated check, right? So this is the data from 2022. There's a good interaction between the treatment that we use at the time of planting and also the drain tile spacing. And look at those bars, the orange bars, right? So whenever we had Inforo Quadris, these are really popping out. So 26 versus 35, right? You got about 9 bags more for the navy beans. The same thing for 25, 40, and 60. So really, the moisture is also important for rhizoctonia, but it's really critical for Aphanomyces, right? So let's say if you have a field that tends to be moist, if you have a drain tile installed, that's going to take care of some of your rhizoctonia problems as well, on the top of everything that you're doing for that. And again, looking at more options for managing rhizoctonia in other crops, our friends from NDSU have this production, the fungicide management guides, please consult this. Like I said, the same chemistries are labeled in dry beans, soybeans, and corn, so talk to your dealer to see what works best for you. So talking about sugar beets, right? We have come a long way thanks to the seed companies. They give us varieties that can yield very high. At the same time, they're able to combine the genetic resistance to rhizoctonia. It's a good example here. Now I have 2 varieties here. The red one is actually kind of the susceptible one, a little bit higher on the rating, close to 5 for rhizoctonia rating. And the blue one is a little bit tolerant to that, right? So the more and more disease we got by the end of the season, we were losing more and more yield there. I think David showed the similar one. We were losing more yield and quality there. But here, if you have a tolerant variety, you're just losing about 2 tons for each point increase in disease rating by the end of the season. With the susceptible one, you're losing about 3.7 tons, right? Pretty soon you're going to lose tons and also quality as well. That's a huge impact. And then seed treatments. Believe it or not, it's been 10 years. It'll be, 2024 season, 10 years we had these seed treatments labeled for rhizoctonia. Number 1, that's Cabina back in 2014, to brand new Zalterra or Excalia as a post-emergence. This is in 2022. But all of these belong to the one class of fungicides we call this succinate dehydrogenase inhibitor. It's a single target. It's not doing any multiple things. It's just inhibiting one key enzyme that's involved in respiration, right? Other thing to keep in mind, right? So fungicide resistance, like how do we deal with that in rhizoctonia? Thank goodness it's not Cercospora. Rhizoctonia tends to complete its life cycle once during the season, so it's not polycyclic. Once it starts infecting beets, when the beets die, it becomes sclerotia, and then it stays dormant. Then the cycle begins next time. So when it comes to risk, it's not as risky compared to the foliar pathogens, but we'll still need to keep an eye. Right? The more exposure it takes over the years, it can develop some tolerance to these fungicides. But so far, we are not seeing any cases. So before I show the data, I think some of the weather pattern that we saw in Crookston, because we inoculated these trials for rhizoctonia, April was fine. And then in May and June, look at that 0.87 and 1.2. It was extremely dry these 2 months. Then the rest of the season was more or less normal. But again, the conditions were really not conducive for developing rhizoctonia in 2023. As we see some of the data, you will understand here, right? So every year we run these 2 different trials. In trial 1, we compare all of the available seed treatments and also fungicides on a susceptible variety that we can find, right? Because if you can manage the disease on a susceptible variety, you can really manage it on a resistant variety, right? And this is kind of apples to apples comparison. So look at the stands here. So by 3 weeks, we got about 230 beets for 100 foot of row, really, perfect one. This is my check, no rhizoctonia seed treatment. We lost some stands. Even by 7 weeks, we were having 210 beets. They're pretty good stands for this. Again, the disease pressure is really low. And looking at the seed treatments, I compared everything in this. They're labeled that I've shown before. Again, everything did very well in 2023. No concern. And statistically, there are no differences among stands, by the 7 weeks after planting. And when I talk about fungicides, right? So we're not just applying fungicides. Sometimes we're also adding starter fertilizers, especially for the soils that you need a little bit of phosphorus boost here. So the way we apply these fungicides, we make 3 gallons of 1034O and then 3 gallons of fungicide. The total 6 gallons is going in the furrow as a dribble, right? But other thing to keep in mind, for you, do your own jar test to see if the fungicide and then the fertilizer that you're using, if they're compatible or not. Then otherwise it's just going to ruin your day when you get in the planter to do that. So looking at the fungicides, again, all of these are actually available right now. And these are also mentioned if you look at the pocket guide, the 2024, all the rates are available in that. Quadris, Asteroid, Elatus, Headline, Preaxter, Proline, and Propols, right? Looking at the data here, again, the blue line that I showed before, that's my non-treated check. We lost a little bit, but not so much. But look at most of the wind furrows, they're slightly tracking above the check here. But again, I said the disease pressure was not as great early on, and the rest of the season, it's more or less normal. But if you don't get enough pressure in the first few weeks, you're always behind. We will never get the disease by the end of the season. And by the end of the season, this is what the rating scale that we use. It's a little different from what David showed. From 1 to 7, this is 0 to 10. Each point is a 10% root rot that you see on the surface, right? So looking at the data between seed treatments as a group and fungicides as a group by the end of the season. So we found slightly higher number of roots, number of harvestable roots for fungicides, 175. A little bit of less disease in terms of severity and number of roots that had the disease, slightly higher yield. But when we look at the ESA, it's comparable to RSA for you. It's about 600 pounds higher, and statistically, it's significant. So the Infurus, even under low disease conditions, did slightly better. We got a little bit of better RSA by the end of the season using this. Now coming on to the post-emergence fungicides. I liked your question, how did they apply the post-emergence fungicide, right? So last few years, we were asking, how are you guys doing? Then 50% of you are saying you're using a band, and other 50% you're doing broadcast applications. So we repeated this trial over 2 years. This is just the data from 2023. So again, some of the fungicides that we used, these are also used as the infurrow. And then the only thing to keep in mind here is the Excalia. This is the only one that's different. If you're using a 7-inch band, you only use 0.64 fluid ounce. And then if it's a broadcast, you have to use 2 fluid ounce. Every other fungicide, it doesn't matter. Whichever way that you use, you stick to those rates for this one. Right here is the data from this particular trial in 2023. All of these fungicides, whether applied as a band or broadcast, and I have this root rot percentage here, right? So very clean root. This is high as like 14. So on a 0 to 10 scale, it's only 1.4, right? This is my non-treated check, just about 8,000 pounds per acre. You see, most of the fungicides, whether you did a band or broadcast, they're looking pretty good. So don't worry. how you apply, but applying the fungicide is the key. But again, when do you apply? Between 4 and 8 leaf application stage, right? So earlier we were tracking 4-inch soil temperatures. When it reaches 65, you're doing it. But right now, just go with your leaf stage. But if you think if your variety is a little bit susceptible to rhizoc, you have a field history, try to get it earlier than later. Shoot for 4 to 6 leaf stage compared to 6 to 8 for that. So this is, again, very useful for you when they post this presentation, right? So rotating fungicide chemistries with different modes of action. But seed treatments, you have no choice. Most of them are SDHI. But between info and then post, let's say if you're using Quadris or Asteroid, you can come back with Excalia because it's a QOI and it's SDHI. I think it's a good practice in general, for managing fungicide resistance. Like I said, it's a little bit of low-risk pathogen for this, but I think it's better to do our job for that. So I'm just going to skip because I covered some of the points, but the other rhizoctonia that we see a few times, I think in the past 9 years, it's only a couple of samples I got. This is called dry rot canker, right? So this is actually one of the fields from southern Maine. It's one of the strip trials. You could see these plants dying later in the season. Typically what you see here, when you dig up those roots, you see these concentric circles, which we don't see with regular rhizoctonia. These are concentric. When you cut them open, you can see these dry pockets. It's almost like potatoes, you saw with the Fusarium pocket rot that's caused in potatoes. This looks very similar. Again, this looks very dry. If you cut this, lengthwise too, you can see the same problem. But the good news, some of the fungicides that we use for regular rhizoctonia should also control this one. We really don't know what are the factors that are really favoring development of dry rot canker. Like I said, it's only a couple of fields here. There are a few instances in Nebraska they found this particular pathogen. But it's a binucleate rhizoctonia. It's a slightly different version of rhizoctonia than the regular rhizoctonia, just to keep under your radar. Now we're going to talk about Fusarium, right? So this is a grower field close to Crookston. Last time they had beets, they thought they were dealing with rhizoctonia. And then next time he was planting beets, he was curious, like most of you, so he planted 2 different varieties in the field. Lo and behold, right, there was only one variety that did very well, other one did not do that well here, right? The problem, it was not rhizoctonia, it was a Fusarium, right? So the only thing we can do right now for Fusarium is actually using a torrent variety. But to do that, we need to know that we are dealing with Fusarium in the field, right? Again, just bring in samples whenever you're in doubt for this one. So what do we have? Traditionally the Fusarium yellows is caused by Fusarium oxysporum. That's very common. But now we do have a Fusarium cicorum. We call this Fusarium yellowing decline. It's as severe as Fusarium oxysporum. But over the years, we also recovered Fusarium equisetae, and there are a few other species over the last 6 years or so. But occasionally, these are 1 or 2 samples in a year, right? Not very common due to this. But I would say predominantly our fields are like 50% of oxysporum and other 50% caused by cicorum. So Fusarium actually does not kill the seedlings before emergence. It's always after emergence, right? These are all done in the pots in the growth chamber about 3 weeks after planting. Again, we use the barley grains infested with Fusarium. We could see these plants dying quickly here. You lose some of the cotyledonary leaves. Again, you have one infected plant. This is all in the growth chamber. But what about in the field, right? Look at the date there. That's June 2nd, 2021. We're seeing the exact same thing. The plants are dying in the field. All of these are pulled from the same field, right? It looks like a little bit of burning because when the plants are young, you don't see the same yellowing symptoms. You have to wait until August. You see those typical half-leaf yellowing symptoms. But this is early on. Sometimes it could be really confused with maybe some kind of burn in the field, right? You could think that maybe herbicide is causing, but you never know. That's why sample these fields. In some of the fields, samples again, look at the traits, like middle of June, the end of June here, 5.7, very susceptible variety, right? The thing with Fusarium, you're not going to see anything on the surface of the roots. They just look clean as whistle. You have to cut those roots, then you see this vascular necrosis because Fusarium is colonizing this xylem, that transports water. So that's how you're dealing with Fusarium. 4.3 variety, end of June here. and then June 30th and July 6th. These are the real samples that are pulled from the fields and sent to our lab for diagnosis, right? So now we are going into August, right? Like I said, typically for Fusarium, excuse me here, that's moving fast here. So again, 4.3 variety here. This is where we are seeing this half-leaf yellowing and then necrosis there. And you cut those roots, you see the vascular necrosis there, right? So this is actually the variety from the same field, but it is 2.5. So we had to cut this like multiple times to recover Fusarium for this, which means, the tolerant variety is doing its job, right? Sometimes it's not uncommon for you to see a little bit of that recovered from the roots for this one. And sometimes it can also be confused for verticillium because it causes very similar symptoms. Half of the leaves turns yellow, but it's more common if you're rotating sugar beets with potato ground, because this is a good pathogen on potatoes. So that can be confused for Fusarium as well. And other thing that looks very similar to Fusarium, this is the genetic chimera. So this is how, this is caused by mutations that happens naturally during the breeding and then seed production areas. So there is nothing to be concerned here, right? So some of the leaves look a little bit yellow, half, sometimes very irregular shapes, and they're not going to cause any damage in terms of yield or anything. It's just the cosmetic thing that you see. So now I want to talk a little bit about the leaf spots, right? So Cercospora, we know a lot about Cercospora, but I think we're seeing a little bit of other leaf spots in the last few years. But again, it's very low frequency, nothing to be concerned about, but something to think about when you see something, right? So Cercospora, clear, very clear, reddish brown, more or less circular spots. But Alternaria looks more or less white, and then they can have some concentric circles. But at the later stage of Alternaria, you see a lot of tattered leaves, and then extended lesions here, right? So when we think about Alternaria, there are really two types that we're dealing with. The ones with the short spores, we call this Alternaria alternata. This is the same thing. You can see the chains of conidia in the culture, or if you take one of these parts and check under the microscope, they look exactly similar. And also the other type of Alternaria, which have, these are the long spores, this is Alternaria brassicae. But most of the times, we are seeing Alternaria alternata whenever we find in some of these field samples. So if you talk to our friends in Michigan, I think they know more about Alternaria than anybody else because, they have a huge outbreaks of Alternaria there. But I think it seems that certain varieties are more susceptible to Alternaria, compared to Cercospora. But thankfully, we don't have any of those issues. And anytime I received a sample that's positive for Alternaria, there was some kind of stress going on in the field, whether it could be drought stress or something happening in the field, or excess moisture. Those beets are not real strong. They're not able to fight everything that's landing on them, so that's where we're picking up Alternaria. And number two, so last year, we had a few instances, growers were applying, early application for Cercospora, and then they did not do anything until later. So the very first and last application. So what's going to happen in between? If you have any type of spots, most of the times these were Alternaria. Again, it's colonizing these parts, even some of the damage caused by ultrablazer was actually colonized by Alternaria, believe it or not. So it's a strong secondary pathogen for that. So how do we manage this? I think most of the fungicides that we have right now for managing Cercospora, these are very good for Alternaria as well. But again, like I said, we don't have the issue with the susceptible varieties in our growing area, right? So now I want to talk about another disease. It's a leaf disease caused by stem phyllium. This is the last one. So talking to our colleagues in Netherlands, back in 2007, they were seeing lots of weird things happening to the beets. It started as like yellow specks, very small. They look like almost manganese deficiency. Pretty soon, the center of the spot, it becomes brown, and then look at the impact by the end of the season. By the middle to end of the August, they lost about 40% of the crop to this particular disease. Now it's widespread. It's called stem phyllium, but the species they found was it's a beticola, stem phyllium beticola. Did we find stem phyllium here in our growing area? Yes, but not that much. In 2019, they found in Michigan, it's a different species. It's very milder version of stem phyllium. In 2021, near Foxholm, Last year I got about 5 samples, but every time I recovered stem phyllium, it's always present along with alternaria. If I looked at 20 spots, there is only one spot that's stem phyllium that's there out of alternaria samples. Again, nothing concerning right now, just keep something under your radar there. So with that, I would like to thank the Sugar Beet R&D Board for funding all this work, and the growers, the cooperatives, and then the industry for providing all the chemicals and supplies, and also the hardworking team behind all of this work. So with that, I would like to thank you, and then Todd, so do you have some time for questions?
Yeah, one question for Ashok?
I'm sorry, I was not able to get the fund. Can you repeat the question? If you're wondering if there are any alternatives for soil-applied fungicides, yeah, I mean, there are some options available in the market, but I think the one that David showed, some of them work in few years and some years they don't work at all. So there's a lot of variability in general looking at the biological products. But I think so far we did not find any effective biological product for rhizoctonia in our trials based on what we evaluated over the years.
All right, in the interest of moving on, we're going to have Ashok step